In Chapter 11, we are going to cover meiosis and sexual reproduction. Now that you have all learned about the process of mitosis and the replication of body cells, where one cell is going to produce two genetically identical daughter cells, we're going to talk about how gametes and sexual cells reproduce. So again, I mentioned this previously. We get 23 chromosomes of information from our mother and 23 chromosomes worth of information from our father. And this happens through the process known as fertilization, when sperm meets egg. So mixing chromosomes from two individuals who are genetically diverse increases the chance of good recombination events to happen. So you've all heard the stories about inbreeding, where cousins are having offspring with cousins or second cousins, and you've heard about the potential for genetic mutations and alterations that occur. Well, the reason that occurs is because the gene pool and the DNA is so similar that recombination events undergo mutations or mishaps. So the more genetically diverse you are from your partner, the better chance you are of having a healthy offspring with minimal mutations, for the most part. So this highly evolved process of sexual reproduction involves the production by parents of two haploid cells and the fusion of two haploid cells to form a single genetically recombined diploid cell. And that will allow you to be the genetically unique organism that you are. So we've talked about the haploid cells, and we've talked about diploid cells, so you should be familiar with those terms by now. The haploid cells that are part of the sexual reproductive cycle are produced by a type of cell division known as meiosis, and that's what we're going to talk about. We're going to make sure that you understand the differences between mitosis and meiosis. So sexual reproduction involves both meiosis and fertilization, and this process introduces variation into the offspring that may account for the evolutionary success of sexual reproduction. The vast amount of eukaryotic organisms, both multicellular and unicellular, can or must employ some form of meiosis and fertilization in order to reproduce. All right, so now let's dive into the actual process of meiosis. So most plants and animals have two sets of chromosomes. And again, you're getting one set from your mother and one set from your father. So that means that animals are diploid, or 2N. The reduction of chromosomes is necessary to avoid doubling in every generation. We saw in the process of mitosis that from one cell, the result after the process of mitosis is two identical daughter cells. So meiosis is going to be a little bit different because we don't want to double in every single generation, and I'm going to explain why on the upcoming slides. Reproductive structures that produce gametes or spores have cells with a single set of chromosome, and these are going to be our haploid cells or our 1N cells. So again, if you're not familiar with the differences between haploid cells and diploid cells, I've explained it here, but also go back to Chapter 10 and review the differences between haploid cells and diploid cells. Meiosis is the nuclear division that forms haploid cells from diploid cells. And this process employs many of the same cellular mechanisms as mitosis. But what's actually happening in meiosis is this process reduces the number of chromosomes by half to produce haploid cells, or 1N cells. So let's briefly recap on the process of mitosis. So we start with this single cell here. We go through interphase and all the interphase checkpoints. Then we enter into prophase, and then prometaphase, metaphase, anaphase, telophase, and then cytokinesis happens and the cell splits, and we result with these two identical daughter cells that have the same genetic information as the cell that we started with. Meiosis is similar, yet it's different. It's going to resemble mitosis, but it actually goes through two rounds of division. So we're going to talk about what happens in meiosis 1 and what happens in meiosis 2. This slide is really helpful to understand what is going on in meiosis 1. So as you can see, a similarity right away between mitosis and meiosis is the fact that they both have interphase. So meiosis is preceded by an interphase consisting of G1, S, and G2 phases, which are nearly identical to the phases preceding mitosis. So same thing in mitosis. Meiosis has the G1, or that first gap phase, and that's focused on cell growth. During the S phase, what's happening is this is going to be the second phase of the interphase, and this is when the cell is going to copy or replicate the DNA of the chromosomes. Really important. Chromosomes are duplicated during interphase, specifically S phase. I always write a question on that. So the chromosomes are duplicated during interphase, specifically in S phase. And then finally in the G2 phase, the second gap phase, the cell undergoes the final preparations to enter into meiosis. So what actually happens when the cell enters into meiosis? We have pretty much the same set of events that happen. We start in prophase, but this one is termed prophase 1. So what's happening is the chromosomes are condensing and the nuclear envelope begins to fragment. The homologous chromosomes bind firmly together along their length, and crossing over is going to occur. Crossing over is going to occur at this place known as the chiasmata, or the cross, and I just circled the chiasmata. The chiasmata form between non-sister chromatids. Okay, spindle fibers emerge from the centrosomes. Then we leave prophase 1 and we enter into prometaphase 1. And what's happening here is the homologous chromosomes are attached to the spindle microtubules at the fused kinetochore shared by the sister chromatids. So here is the centromere with the kinetochore. And the chromosomes are continuously condensing, and the nuclear envelope completely dissociates and disappears at this phase. Then we enter into metaphase 1. So remember, what happens in metaphase? The chromosomes are going to line up along the metaphase plate. So you can see the chromosomes, they kind of align in the middle, getting ready for the dissociation events to happen. So they maneuver to the middle as they are governed by the microtubules, moving them around. And then we enter into anaphase 1. So anaphase is the beginning of the actual separation. The spindle microtubules pull the homologous chromosomes apart, but really important here, the sister chromatids are still attached at the centromere. So the homologous chromosomes separate, but the sister chromatids remain attached. And then we get into telophase and cytokinesis. So the sister chromatids in telophase 1, they arrive at the poles and they begin to decondense. And then what's going to happen is that cleavage furrow is going to form. And that's going to divide the cytoplasm. And as the cytoplasm divides and the cell splits, we get a result of 2 haploid cells. And each cell is going to contain 1 duplicated copy of each homologous chromosome pair. So I explained this on the previous slide, but again, this concept and all the concepts that I reiterate are very important, and when I reiterate concepts, you're most likely going to see them on the quizzes or exam. So again, a major similarity between meiosis 1 and mitosis is that they both have interphase. They both have G1, S phase, and G2, and those checkpoints. Chromosome replication in S phase makes identical sister chromatids. So this is where the chromosomal replication takes place. Again, I mentioned this again because it's very important. Chromosomal replication in S phase of interphase. And the sister chromatids remain attached at the centromere. So again, this is going to be the centromeric region towards the center where the sister chromatids link up. The kinetochore is this piece right here. And again, they're held together by these mitotic spindles and the microtubules. The microtubules will also play a role in governing the movement of the sister chromatids toward the metaphyseal plate, or the metaphase plate, when the stage of metaphase is necessary. So I know the slide with the entire process of meiosis 1 may seem a little overwhelming, so that's why I'm going through each one of the phases individually so that you understand and hear all of this information again. So we've left interphase and now we are in prophase. Homologous chromosomes pair and the synaptonemal complex holds them close together in synapsis. So the synaptonemal complex is a lattice of proteins between the homologous chromosomes. And this first forms at specific locations and then spreads outward to cover the entire length of the chromosome. The tight pairing of the homologous chromosomes is the synapsis. And in synapsis, the genes on the chromatids of the homologous chromosomes are aligned precisely with each other. The synaptonemal complex supports the exchange of chromosomal segments between homologous non-sister chromatids, and this is done through a process known as crossing over. So this is where information or the segments of the chromosomes are exchanged through this process known as crossing over. Visible structures at crossover points are called chiasmata, and the 4 chromatids held together by a chiasmata are called a tetrad. So as I mentioned earlier, the process of crossing over is responsible for the exchange of genetic material. And what we show here on this slide is the process of crossing over that's occurring between non-sister chromatids of homologous chromosomes. So this chromosome here and this chromosome here are homologous to one another. So what happens after they align? Then crossing over occurs. You can see the crossover event right here. And then the recombinant chromosomes are the ones that are actually exchanging the genetic information. So you can see this one here and this one here. They are considered to be the recombinant chromosomes. Again, here and here. So now you see this red piece in the blue chromosome, and you see this blue piece in the red chromosome. The segment of the chromosomes that do not exchange genetic information, they are known as non-recombinant chromosomes. So again, this piece here and this piece here are non-recombinant chromosomes because they're not exchanging genetic material or genetic information. So these are the 3 events that you need to know that occur in prometaphase 1. So first, the spindle fiber microtubules, they're going to attach to that kinetochore and the proteins associated with the kinetochore at the centromeres. The homologous chromosomes are still going to be held together at the chiasmata, or that centerpiece. And in prometaphase 1, I always ask this question down here, at what stage of meiosis 1 is the nuclear membrane completely broken down? And that is occurring in prometaphase 1, complete dissociation of the nuclear membrane. And then in metaphase 1, what's happening is the homologous chromosomes, they are going to be arranged at the cell equator. They're going to align on that metaphase plate, and their kinetochores are going to be facing opposite poles. So both the maternal and paternal chromatids orient randomly, and they're mixed when they migrate to the poles. And this mixing is responsible for creating additional genetic variation among daughter cells. And this additional genetic variation is very good for proper recombination events to happen. And then in anaphase, the microtubules are going to be responsible for pulling the tetrads apart. And the chiasmata are broken, but the sister chromatids, really important here, the sister chromatids are going to remain attached at the centromere. In telophase 1, the separated chromosomes arrive at opposite poles. And the remainder of the typical telophase events may or may not occur depending on the species. In some organisms, the chromosomes actually decondense and the nuclear envelopes form around the separated sets of chromatids produced during telophase 1. In other organisms, cytokinesis, or the physical separation of the cytoplasmic components into daughter cells, occurs without the reformation of the nuclei. In virtually all species of animals and in some fungi, cytokinesis is responsible for separating the cell contents through the means of a cleavage furrow, and I showed that on the slide with the picture of the whole process of meiosis 1. In plants, we've talked about this in chapter 10, a cell plate is formed during cytokinesis by the Golgi vesicles fusing at the metaphase plate. This cell plate will ultimately lead to the formation of cell walls that separate the two daughter cells in plant reproduction. This is a really cool meiosis animation, so at this point please pause the PowerPoint and click on this link and check out this video. Alright, now we are moving into meiosis 2, but before we talk about meiosis 2, I want you to be aware that in some species, cells actually enter into another brief interphase known as interkinesis before entering into meiosis 2. Really important about interkinesis is it lacks an S phase, so the chromosomes are not duplicated. I always write that as a test question. Why are the chromosomes not duplicated in interkinesis? It's because it lacks an S phase, so they cannot be duplicated. They cannot go through DNA synthesis. The two cells produced in meiosis 1 go through the events of meiosis 2 in synchrony. So in meiosis 2, the sister chromatids within the two daughter cells are going to separate, and they're going to form 4 new haploid gametes, which you see down here, once the process of cytokinesis does occur. The mechanisms of meiosis 2 are very similar to mitosis, except that each dividing cell has only one set of homologous chromosomes, each with 2 chromatids. So each cell has half the number of sister chromatids to separate out as a diploid cell undergoing mitosis. In terms of chromosomal content, cells at the start of meiosis 2 are similar to the haploid cells in G2 that are preparing to undergo mitosis. And I'm not going to read through each one of the events here. So again, same set of events that you see in mitosis and meiosis 1. We go from prophase to prometaphase to metaphase to anaphase, telophase, and cytokinesis. But because this is meiosis 2, there is a 2 after each one of the phases. So we have prophase 2, prometaphase 2, metaphase 2, anaphase 2, telophase 2, and just cytokinesis again, the separation of the cytoplasmic components. So remember, I gave you some different types of events that could happen in telophase 1. So the chromosomes could have the potential to decondense. And if they did decondense in telophase 1, what's going to happen in prophase 2 is they're going to condense again. If the nuclear envelopes were formed, they're going to fragment into vesicles. And then the MTOCs, or the microtubule organizing centers that were duplicated during interkinesis, they're going to move away from each other toward the opposite poles, and new spindles are going to be formed. In prometaphase 2, the nuclear envelopes are completely broken down and the spindle is fully formed. Each sister chromatid is going to form an individual kinetochore that attaches to microtubules from opposite poles. And in metaphase 2 and anaphase 2, these processes are very similar from the process that we have already talked about. In metaphase 2, the sister chromatids are maximally condensed and they are going to align at the equator of the cell. In anaphase 2, the sister chromatids are pulled apart by the kinetochore microtubules and they're going to move toward opposite poles. Non-kinetochore microtubules are going to be responsible for elongating the cell. In telophase 2, the chromosomes are going to arrive at opposite poles and they are going to begin to decondense. The nuclear envelopes are going to form around the chromosomes, and if the parent cell was diploid, which is the most common case, then cytokinesis is now going to be responsible for separating the two cells into 4 unique haploid cells. The cells produced are genetically unique because of the random assortment of paternal and maternal homologs and because of the recombination of maternal and paternal segments of chromosomes that occurs during the process of crossing over. So now let's compare mitosis and meiosis. So they're both forms of division of the nucleus in eukaryotic cells, and they do share some similarities, but there are also a number of differences that lead to very different outcomes, and these outcomes are shown in the picture here. So again, the most important differences are listed here in the text, and also I want you to understand what's happening in this representative picture. So again, mitosis is a single nuclear division that results in two nuclei that are usually partitioned into two new cells. The nuclei resulting from a mitotic division are genetically identical to the original nucleus. They have the same number of sets of chromosomes, one set in the case of haploid cells, and two sets in the case of diploid cells. In contrast, meiosis consists of two nuclear divisions resulting in four nuclei that are usually partitioned into four new genetically distinct cells. The four new nuclei produced during meiosis are not genetically identical, and they contain one chromosome set only. This is half the number of chromosome sets in the original cell, which is diploid. And the main difference between mitosis and meiosis occurs in meiosis I, which is a very different nuclear division than mitosis. So remember, in meiosis I, what's actually happening is the homologous chromosome pairs physically meet, and they're bound together with the synaptonemal complex. After this, the chromosomes develop chiasmata, and they undergo crossover between non-sister chromatids. And in the end, the chromosomes line up along the metaphase plate as tetrads, with the kinetochore fibers from opposite spindle poles attached to each kinetochore of a homologue to form a tetrad. And all of these events that I just talked about only occur in meiosis I. One of the main purposes for the existence of organisms is to reproduce, to pass your genetic code on to your offspring. And again, we've all heard of survival of the fittest. The best genes are going to become dominant over time in order to ensure survival of the species. And if the genes do not get passed on, the best genes do not get passed on, well then the species may become extinct. So we're going to talk about the three main categories of life cycles in multicellular organisms. We're going to talk about diploid-dominant, haploid-dominant, and what it means for there to be an alternation of generations. Most animals have a diploid-dominant life cycle strategy. The only haploid cells produced are the gametes, or the sex cells. The germ cells produced by the gonads and which are the testes and the ovaries, they use mitosis to perpetuate the cell line and meiosis to produce the gametes. There is no multicellular haploid life stage, and fertilization occurs when two gametes fuse to restore the diploid state. Fertilization, sperm meets egg. Most fungi and algae, or the body of these organisms, which in fungi we don't really see the body of the organism until the fungi is about to die. So after a rainy day or a rainy morning, you may go out in your front yard and you may see some mushrooms popping up. Well, if you didn't know this already, mushrooms spend the vast majority of their life underground, and they only come up to the surface during the end of their life cycle, or right before they're about to die. So this is known, or fungi reproduce via the haploid-dominant life cycle. And it's ecologically important, and this is also haploid. So during sexual reproduction, specialized haploid cells from individuals designated the positive and the negative mating types join to form a diploid zygote, which immediately undergoes meiosis to form four haploid spores. Although haploid like the parents, these spores contain a new genetic combination from two parents. The spores form multicellular haploid structures by many rounds of mitosis. So we've talked about the haploid and the diploid life cycles. Now let's talk about the third life cycle, which is used by some algae and all plants. It's actually a blend or a mix of the haploid-dominant and diploid-dominant extremes. Species with alternation of generations have both haploid and diploid multicellular organisms as part of their life cycles. The haploid multicellular plants are called gametophytes. So we see the gametophytes here. Also laid out there for you. And they're called gametophytes because they produce gametes from specialized cells. Meiosis is not directly involved in the production of gametes in this case because the organism that produces the gametes is already haploid. Fertilization between the gametes forms a diploid zygote, and the zygote is going to undergo many rounds of mitosis, and this process is going to give rise to a diploid multicellular plant called a sporophyte. Specialized cells of the sporophyte will undergo meiosis, and they are going to produce haploid spores, which you see right here. The spores will subsequently develop into the gametophytes after they germinate, and this process continues over and over again.